Welcome to our biology lessons with Dr. Hanaa. Today we will solve with each other June 2025, variant 2, paper 6. Question 1. A student investigated the effect of concentration of salt solution on osmosis in potato dishes. So when we find the word of effect of, we have to know that after this word, so we have independent variable or changed variable. So when he asks about what is independent variable or what is the changed variable, so we say this concentration of salt solution on osmosis in potato tissue. And this is we have to know, or we have to be measurable, and we have to know now what is the dependent variable. The student used 3 different concentrations of salt solution and potato tissue that had been cut into cylinders. So we have cut, this is a kind of hazard. Using the information in the table, decide the volume of S and W needed to make 50 cubic centimeter of 0.5 mol per decimeter cube salt solution. Complete table 1.1 by writing in the volume of S and W needed to make 0.5 mol per decimeter cube salt solution. Right. Firstly, we have to concentrate about the volume of 1.0 mol per decimeter cube, the salt solution, S in cubic centimeter, and this is W for the distilled water also by cubic centimeter. And we put 50 cubic centimeter of salt solution plus 0 of distilled water, so the total is 50. And he wants us to calculate the concentration of 0.5 and how cubic centimeter of salt solution and how many cubic centimeters of distilled water added to make the half of the concentration. So if you have to know that this is 1 and 0.5, this is the half of it. How can we make it diluted to the half? So we will take 25 cubic centimeter of salt solution with a 25 cubic centimeter of distilled water to make the total of 50 cubic centimeter of the total volume, and with a half concentration of the salt solution at the end, which is the final one. And the last one, which is 0 salt solution plus 50, which is equal 50. As he said it, the total volume of solution, this is the final volume of the solution, which is 50. This is one of the controlled variables. So if you concentrate well, we can find S3, 0 salt solution, and 50 distilled water. So we can use S3 as a control. What's meant by control? So nothing that affect the reaction, and just put it a distilled water, so use it as a control to compare the other result. The student used this method. So step number 1, label 3 beakers, S1, S2, and S3. Use the volume of 1 mol per decimeter cube salt solution S and distilled water W shown in the table to make the salt solution in the beaker S1, S2, and S3. Put the potato cylinder onto the white tile. What is a white tile? It looks like a ceramic piece. And cut each potato cylinder into 3 millimeter discs. As shown in the figure, just bring the cylinder of potato and just make it a small discs. Each one of them 3 millimeter. And we have to cut. This is a kind of, this is hazard. Place 10 potato discs in a line next to a ruler as shown in figure 1.2, and then measure the total length of 10 potato discs. Put it beside each other, then measure the total distance of the 10 potato discs. Put the 10 potato discs into beaker S1. Repeat step 4 to 6 with the other potato discs and salt solution in beaker S2, which is 50% or 0.5. and S3, which is considered as a control. Start the stop clock and leave the potato disc in the beakers of salt solution for 20 minutes. So this is a controlled variable. Remove the 10 potato discs from a beaker S1 and put them onto the white tile. Line them use along the ruler and measure them again. The final total length of 10 potato discs. Repeat step 9 and step 10 with the potato discs in beaker S2 and S3. They give us this note. So beaker S1 started with a total length of 102, and after 20 minutes was 94. S2 was 103 and becomes 98. S3 started with 100. Figure 1.4 is a diagram showing the actual size of 10 potato discs from a beaker S3 after 20 minutes. So we have to bring the ruler and just measure the distance, the total distance, and we have to convert it into millimeter. So if your measurement, this is, for example, 10.2, so we have to convert it by, multiply it by 10, because 1 centimeter equals 10 millimeter. So it was 10.2 and multiply 10, so it is equal 102. Prepare a table and record the result shown in figure 1.3 and your measurement in 1AII. Include the final concentration of salt solution in your table. So now we have many points. You have to put beaker, so we have S1, S2, and S3. And final concentration that we have it, this one, we have to put it 1.0, 0.5, 0.0, and the initial and the final length of 10 tests. So we have the beaker S1, S2, and S3. The final concentration 1.0, 0.5, and 0.0. So why we write it by this way? Because we have to write it with the same decimal places. Initial length 102 and it becomes 94. This is from the sticky note that give it to us. And the initial one for S2 103 and the final 98. And the initial length for S3 100 and becomes 102. Calculate the change in total length of 10 potato discs in each beaker. So if you have this is initial length in millimeter and this is the final one, how can we calculate the change? And maybe this change may be in a positive and maybe in negative, maybe increases and maybe decreases. So we can calculate the change final minus initial. For S1, final, which is 94, minus 102, which is equal negative 8. This means that it decreases by 8. What about S2? So final minus the initial. So 98 minus 103, which is equal negative 5. So this means that it decreases by 5. What about S3? So final minus initial, it will equal 102 minus 100. So it gives us positive 2. This means that it increases by 2. So this is my answer, the beaker S1, negative 8 millimeters. So it decreases by 8. S2, negative 5. So it means that it decreases by 5. And S3 plus 2, it means that it increases by 2. I know that in the mark scheme, they just wrote the value, the answer directly. But I mean that if we have the word of change, if I have the word of change, so this change may be in a positive, this means that increases, maybe in negative. So I think that's we have to put in consideration this is increases or decrease, the change by increasing the value or decreasing it. State the conclusion for the result of this investigation. So we have to return back to the objective of the experiment. you have to know that the effect of concentration on that, the osmosis, this is the movement of water. So the higher the concentration, the more water moves out of potato discs. What about the dependent variable? So we measure what? The length of potato. So we have, this is dependent variable, the length of potato discs. Suggest why the student used 10 potato discs instead of 1 potato disc in each salt solution. We know that if you just have 1 potato disc, it is hardly to measure it. So we have to get a representative data. So we have to increase the sample to be easily measured. So to get representative data, as the change in 1 disc is too small, it is too hard to be measured. So at least we have to measure with 10 potato discs. Describe one safety precaution that the student should take when preparing the potato discs. So we know that this is they use a knife. Okay, so the cut, you have to cut on using a cutting board, away from hand by using cutting board, or cut on a flat surface. The student added a few drops of yellow-brown solution to test one of the potato discs. The solution changed to blue-black color. Identify the solution added and state the conclusion for this test. So what about this solution? Yes, so it was yellow-brown, okay, and it becomes a blue-black. So this solution is sure, yes, iodine solution. This means that the potato discs has starch. Next question in front of you, a leaf from a potato plant. So it's, I prefer بصراحة all the leaves to be drawn, this easily one. So we have a technique, you have to make analysis for leaf. Started with the midrib, we can find it in the middle. It is better to start with an easy point to be sure that your length is correct. And the midrib should be in a double line because it is thick. And then you have to concentrate about the details. We have 2 details here. So you have to know this is the pointed one, pointed tip. And also, so this is very important part to be pointed. And we have here other details. This curve, and then it looks like a spine. Looks like a spine, another one, and this curve. So we have 2 main details you have to put in a consideration when we draw the leaf. And then you have to make the outline to print this image in your brain. Okay. And the other side. Okay. And then we have small veins. They never start from the same point. So they started by alternate, not from the same point. And then I let you to see my drawing at the end. So you have to draw it large in size. This one mark for the diagram size, and the second one for the outline, not be feathery drawing, not be broken line, and 2 other details about the diagram. This is my drawing about the details that I need. Line PQ represent the length of the leaf. Measure the length of line PQ. So my length was 6.3 centimeter, but we have to convert centimeter into millimeter. So 1 centimeter equals 10 millimeter. So when we convert it, so 6.3 multiply 10, which is equal 63. And then the actual length of this leaf is 39. use the measurement and formula to calculate the magnification. And they said, and help us by giving us the rule. And also we have a magic triangle, which is I am. I am, so I, this is related to the image, A related to the actual length, and M related to the magnification. If you want the magnification, so we have to put the image divided the actual length. 63 divided 39, which is equal 1.615. And they need the answer with the two significant figure. So just 1 and 6. So, and what about this after 6? So we can find it 1. So we would ignore it. So 1.6 times. Figure 1.6 shows a leaf from a tomato plant. The tomato leaf is narrower than the potato leaf shown. So compare the leaves shown in figure 1.5 and 1.6. Describe one visible difference and other than size and one visible similarity. So if we concentrate well, we can find this. We have here many loops. We have many loops here, but the other one, there is no loops. So potato leaf is a single, not looped, but tomato leaf has loops. But both have veins, both have leaf stalk. Where is the stalk? This one. So both have leaf stalk and both have pointed tips. This pointed one, and we drew the in the previous figure pointed tip. The enzyme pectinase is used in a production of apple juice. In an experiment, students investigated the production of apple juice using 5 different concentrations of pectinase solution. For each concentration, the student put 100 grams of crushed apple in a beaker, added 10 cubic centimeter of pectinase solution to the crushed apple. So we have here this 100, this is the mass of apple, this is controlled variable, and we have 10 cubic centimeter of pectinase, this is also controlled variable. Left the crushed apple and enzyme mixture for 30 minutes at 40 degrees Celsius. Also controlled variable. Filter the apple juice from the mixture, measure the volume of apple juice production. So this is dependent variable. The temperature was maintained at 40 degrees Celsius. Describe how the student could maintain the crushed apple and enzyme mixture at the constant temperature. How can we maintain the temperature? So we can use thermostatically controlled water bath. Or also you can write incubator. So incubator is right. So you can write it thermostatically controlled water bath or incubator to be kept at 40 degrees Celsius. State 2 other variables that were kept constant. That just wrote it here. We have the mass of apple, the crushed apple, so its mass, and also we have the volume of pectinase, which is 10 cubic centimeter, and the duration time left, which is 30 minutes. The result of this investigation are shown in the table. The percentage concentration of pectinase solution and the volume of apple juice production. So when we have any table, we have to know that the first column for the independent variable, this is independent variable, and we have to put it on the x-axis. And what about the other column? This is dependent variable, and we write it on the y-axis. And you have to put in a concentration, the least value and the highest one, least value and the highest one, and we have here level of or plateau, or the volume at a certain concentration does not change. So when we start to draw the graph, we have to put 4 points because the graph with the 4 marks. The first one, which is axis label. So if you concentrate well, we can find 4 marks. The first one for the axis label. Just write at the heading of the table as it is. So we have the percentage concentration of the pectinase solution on the x-axis. And on the y-axis, volume of apple juice produced per cubic centimeter as it is. If you just write them as they are, so you will take the first mark. Then what about the second one? So we have to make the scale. You have to look to the least and highest values and then count how many big boxes and try to distribute these values to fill all the space provided. So I divided the percentage concentration 0, 0.5, 1, 1.5, and 2. And what about the volume? It started from 0 and by increasing 5, 5, 5. So 0, 5, 10, 15, 20, 25, and 30. So if you're just making a correct scale, so you'll take other mark. So you have to occupy the graph provided with the use 2/3 of the graph paper. What about the third mark? So we have to plot the point. How can we plot the point? So by using X. So in front of us, two options, maybe X, maybe dot with a circle, but which one of them is better if we have just only one plotting values? So you just use X. Now, when we start to use dot with a circle, if we have a key, so if we have different values of different data, so and we have to draw all of these data on the graph paper, so we can use X and dot with a circle. But if it is one, you just use X. So if you just after plotting, so the third mark. Then we have to use a pencil and ruler. We try to join all the points with each other. It's okay in biology to join them from dot to dot. So it's okay in biology from dot to dot using pencil and ruler. So this is line. So this is the last mark. Use the graph to estimate the volume of apple juice produced with the 0.5% pectinase solution. So we have to return back to the graph. And you have to go to the concentration 0.5 and go with the intersection of the line and move horizontally to the x-axis to reach to the value on the y-axis, which is the volume of apple juice produced per cubic centimeter, and we can find it for T. And you have to show your work on the graph paper. Describe the effect of pectinase solution concentration on the apple juice production. Yes, as the concentration of pectinase solution increases, so we can find that the volume of apple juice produced increases, then level off, or then the volume does not change. So you have to write the whole description of the graph. So as the concentration of pectinase solution increases, the apple juice production increases, then level off. And we know that at the value 22, as we said it before in the table. Pectinase is used to produce apple juice commercially. Suggest what extra data would be required to determine if 0.8% pectinase solution is the optimum concentration for the production of apple juice. That if you'd like to know this is an optimum or not, so we have to take small intervals around the 0.8 to be sure that in the 0.8, which is the optimum one. Maybe yes and maybe no. So measure more concentrations of pectinase around the 0.8% using small intervals as that around the 0.8, 0.6, 0.7. This is before 0.8. And after 0.8, we can use 0.9 and 1 to be sure that this is the optimum value or not. Draw and label the apparatus that student could use to filter and measure the volume of apple juice produced. So we have to draw something that filter and something that measure the volume of liquid. So when we use something to filter anything, so we can use filter paper to be standard on the filter funnel, okay? And how can we measure the volume of apple juice? In front of you, two options. You can draw a measuring cylinder or you can draw conical flask. So here, so in front of you, this is measuring cylinder and using funnel And inside the funnel, you have to put the filter paper. And don't forget to make the graduation on measuring cylinder that indicates it's able to measure the volume of apple juice produced. And also don't forget to label the diagram. Hydrogen carbonate indicator can be used to determine the concentration of carbon dioxide in a solution. The table shows that the color of indicator at different concentration of carbon dioxide. So we have 3 different concentration, maybe high, maybe medium, maybe low. And the color of hydrogen carbonate indicator at higher concentration of carbon dioxide, so it becomes yellow. And in a medium concentration of carbon dioxide, it will be red. And if there is low concentration or no concentration of carbon dioxide, it will be purple. Aquatic plants photosynthesize and use of carbon dioxide when in the light. Aquatic plants respire and produce carbon dioxide in the light and in the dark. When the carbon dioxide produced is equal to carbon dioxide used, so the hydrogen carbonate indicator will be red. So this means that in medium concentration of carbon dioxide. Use an aquatic plant, plan an investigation to determine the light intensity at which the rate of photosynthesis is equal to the rate of respiration. That we will use a hydrogen carbonate indicator and the color will be red. So who can tell me what is the independent variable here? So we have to know that to determine the light intensity that or at which. So this is independent variable, which is the changed variable. How can we change light intensity? In front of us, 2 different ways. You can change the distance between light source and the aquatic plant, or we can use different sources of light. For example, light bulb with the different intensities. Different sources of light that change light intensity or change the distance between the light source and aquatic plant, at which the rate of photosynthesis is equal to the rate of respiration. So we will use here the hydrogen carbonate indicator, but the problem here is this is judged by eye. So this is subjective. How can we improve it? Yes, by using colorimeter or color chart. So let's start this investigation step by step. Firstly, we have to bring 3 test tubes, put in each aquatic plant of the same species, same type, same size, and same age. So this is some of the controlled variable. Then put in each same volume of water, also controlled variable, and keep the plant underwater. Keep the test tube in thermostatically controlled water bath at 30 degrees Celsius. Also, this is controlled variable and should be maintained using thermostatically controlled water bath, or we can use incubator. So both are correct. Put the test tubes at different distances, 10 centimeter, 20 centimeter, 30 centimeter from a light source of same intensity. So we have just only one factor, which is the distance. So we consider this point, which is independent variable. And then when we explain this investigation, we have to keep the temperature constant. So we said at the 30 degrees Celsius. And what about the effect of source of light? Maybe increase the temperature. So we have to use heat shield to prevent overheating done by light source. So we have to use heat shield to prevent overheating done by light source. Leave the aquatic plant totally submerged for 5 minutes for equilibrium. What's meant by equilibrium? This means that all the components of this experiment start to work together. And then you can leave the experiment for 30 minutes, for example. You can write it as 60 minutes. So the time that you want, this is also controlled variable, and this is also controlled variable. And then we have to take a sample from each test tube and using a hydrogen carbonate indicator by dropping using a syringe to observe the color change. But we have to improve it by using colorimeter or color chart to observe the color change that becomes red. This is at the end. This is the dependent variable. Okay, and with its improvement, you have to repeat it at least 3 times for each distance and take an average. And finally, with the safety precaution, you have to wear gloves, goggles, and use tongs. So I hope that you enjoy the time of solving the exam. Waiting for new exams with each other. Don't forget to like, share, and subscribe. Thank you and goodbye.